So we're next going to be joined online by Dr. Tom Monath, who's the Chief Scientific Officer at Crozet Biopharma. Dr. Monath. Presto. Dr. Monath?
Sorry, I was on mute. My fault.
There you go. There you go.
Good afternoon, everybody. I assume you can hear me now.
Yes, sir.
Good. And am I to share my slides, or are you advancing them there?
You are.
Okay.
He's doing it, right?
And you can see it, all right?
Affirmative.
Good. All right, well, I'm going to talk about, really give you a conceptual idea about how one might go about developing a live attenuated vaccine that you've heard from Lisa Schubitz and is in development as a dog vaccine. I am a vaccine developer and been focused mainly on viral zoonotic vaccines and brought 5 through to FDA approval. And I got very intrigued with this organism after talking to friends who are developing the dog vaccine. And it's a wonderful One Health story. So my disclosure is that I'm working with the company Crozet Biopharma that's interested, but does not have funding currently to work on the vaccine. And if we do, we'd be very happy to advance it. So I think you've heard that a vaccine is feasible because natural infection presumably protects against subsequent disease. And the canine vaccine is in advanced development and has a favorable expectation of getting through commercial success. The same vaccine is likely to be safe and effective for humans. The dog is an adequate IND-enabling animal model. And the problem is funding and policy rather than technical risk, in our opinion. Now, you've heard about the Delta CPS1 vaccine, which was developed by John Galgiani and his colleagues. It's genetically engineered C. possidacea with a deletion of the CPS1, which is a virulence factor and is critical for circulation and propagation of the parasitic phase. So it's very much like other live vaccines that create a abortive infection and present multiple antigens mimicking the natural infection to the host. Now, we have many approved vaccines for humans. The majority actually are live attenuated vaccines, both viral and bacterial. And more recently, some of these are genetically modified organisms using transgenic approaches. So they're kind of modern vaccines. The older live attenuated vaccines were developed empirically by passing the agent in an unnatural host. But these are very successful vaccines. They generally give you long-lasting immunity, often after a single dose. And of course, generate good B and T cell responses to the organism and are self-adjuvanting. You've heard that this live attenuated vaccine works well in the mouse model, high-grade survival, even in animals, skid mice or animals with other immunodeficiencies. And appear to work well in the dogs as well, reduction clinical score and fungal burden. And I think pretty clear that you need good T-cell immunity for vaccine to work, and that's presumably the basis for protection in both mice and dogs. So what are some of the challenges to developing a human vaccine? One that's always brought up is manufacturing, because most contract manufacturing organizations that make various kinds of biological products don't like spore formers. They take pains to make sure that they don't enter the facility. And so that's an issue, and I'll come back to that in a minute. On the preclinical side, I think one thing that's clearly missing and will be required from a regulatory perspective is a biodistribution study. I think that probably needs to be done in non-human primates, and I'm glad to hear the progress on the model that Deb Fuller described in pigtailed macaques. It's important to have an immunological marker, a biomarker of immunization. Clearly, that's going to require T-cell assays, but it's certainly feasible. And the question of that's going to be the fundamental question in human trials where you're dependent on an immunological marker to know whether you've done anything, at least in phase 1 and phase 2. And in phase 3, of course, it's going to be necessary, I think, to show protection against the disease. This is the first live eukaryotic vaccine. I think that raises safety concerns and may require a large safety database in the clinic, but that's fairly clear. And on the policy side, the risk is going to be, what is the Advisory Committee on Immunization Practices going to recommend? They usually require some pharmacoeconomic data that seems to be pretty clear now from this meeting that shouldn't be an issue. So what about manufacturing? Well, there turns out to be one large company, it's in the Boston area, that is working with spore formers, over 150 different spore-forming bacteria and fungi. They're working on the microbiome. They're working with many big pharmaceutical companies. They have beautiful facilities and could take this vaccine from clinical trial material all the way through commercialization. They're very excited about the project. Had multiple discussions with them. And I think, some of the things that need to be done on the manufacturing side, you can't do sterile filtration of this product as an injectable, so it has to be an aseptic process. It's currently an open manufacturing system. I'd like to develop a bioreactor closed system in liquid media. Probably some additional downstream purification required from the dog vaccine. And it will be presented as a liquid frozen product at -70. As you've heard, this is not a problem. It's been done with the Ebola vaccine, it's been done with COVID vaccines, and doesn't present an issue. So we're quite confident the manufacturing is achievable. I mentioned the IND enabling non-clinical studies, biodistribution, probably in monkeys, repeat dose tox study. I think that could be done in mice, and probably a study either in mice or in rabbits showing safety with respect to the brain and spinal cord. Now, I'm going to walk you through a clinical development plan. This will change ultimately, but it's just a conceptual idea. I think you start with phase 1, as you always do. It would be a dose response study, small numbers of subjects. We've got good guidance from the dog data that probably 10,000 to 30,000 spores will be adequate. We'll have to have an immunological assay, obviously, to measure that. Placebo-controlled study. I think we'll include an IM arm also because it may be more likely to avoid some of the local reactogenicity that, even though it seems to be well tolerated in dogs, often that can be avoided by the IM route. So we'd look at that. Phase 2 would just be an expanded study in people who were both skin test negative and positive. I think it would be very important to establish safety because you're not going to screen people for immunity to this disease before you vaccinate them at a population level. And so we would have some dose guidance from phase 1, look at a high and low dose in both negative and positive people, recognizing that the skin test is relatively insensitive and could use other measures of immunity as well, screen. So that's all fairly straightforward. The hard part is getting to a pivotal study to get FDA approval. There are three ways you could do this with vaccines. The traditional pathway is to demonstrate efficacy in randomized controlled study, reduction in disease incidence. The accelerated approval strategy allows you to use a biomarker, which is predictive of efficacy. I don't think that's going to be possible here. And anyway, it's not final licensure. You have to do an effectiveness study later. And then the animal rule. I don't think we have a stringent enough model in which to use the animal rule. As long as it's achievable to do a pivotal study by the traditional pathway, I don't think you would be allowed to use the animal rule anyway. What are the regulatory considerations? Well, obviously you have to recognize the disease, the efficiency in collecting and recognizing an illness, fever, cough, chest pain, and then have sufficient diagnostic precision to diagnose cocci. These are challenging, but I don't think, I've heard a lot in this meeting. It's very encouraging that on the diagnostic side. So I think those are manageable. One trial or more. Now, typically the FDA will want, if you're going to rely on one study, you're going to have to show very strong evidence, such as a high lower bound of the 95% confidence interval. For COVID, FDA knew that they may have to approve based on one study, so they set the bar pretty high with a lower bound of 30%. That may not be feasible with cocci. So I think 2 phase 3 studies are going to be required. I'll get to that in a minute. Can you stack the deck by eliminating immune subjects? Royce Johnson told us the difficulties there. It's, the idea that no second infections occur relies on old epidemiological data. Is that going to be accepted by the agency? And I think there'll be concerns about bias if you try to focus on susceptibles, even if you could identify them. And then, of course, ultimately, there are these special populations that you would like to address with a vaccine. And so that raises other concerns down the line for clinical development. So is the disease incidence sufficiently well defined? Well, there are multiple studies, and we've heard more today. They're pretty consistent, actually, with about 1 to 2 per 1,000. And this is in the general population. These are reported, recognized, diagnosed cases, and obviously, we heard that maybe 33 times underreported. So that's actually, those data from CDC are quite interesting because I think if you look carefully at your clinical population, you're not going to have that 33 times underreporting rate in the clinical trial population. So, but I think I think we have to power the phase 3 study based on these data, not select for non-immunes, but show that the vaccine works in the general population. Now, talk about 2 different trials. One could be a study in a very high-risk group. The California state prisons are obviously interesting. You heard about that today or yesterday. And they could be skin test negative selected. The incidence, it may have dropped a bit from the older published paper, but it's pretty high, 2 to 4%. So a fairly modest study here, powered based on the data you see here, could get a signal of efficacy. So that would be very interesting, but it doesn't, I think from a regulatory perspective, you need more. This could be one of the 2 pivotal studies. these are the older data. I guess the incidence in Pleasant Valley and Avenal that we heard has dropped considerably, but it's still fairly high. Certainly ethical to do a clinical study in prisoners because the basic rationale is that they're more severely impacted by the disease you're testing than the general population. There may be other populations too. The larger study would require, done in the general population would be the pivotal study. You do your lot consistency, 3 different lots represented, hopefully some immunological marker that is working well, and would use sites that are generally high incidence. Whether or not you would do skin test or other immunological selection is something we would have to discuss with the agency. I think it'd be better not to, but it certainly would maybe help select a more vulnerable population. And the assumed attack rate, 0.25%, is conservative based on the data I showed you before. It's still a modest trial, 5,000 something. We do vaccine trials sometimes require 100, 200,000 people. So challenging, expensive, yes, but doable. So I'm not going to dwell on this. We've mapped out a strategy for development, as you do in industry. A lot of the preclinical exploratory work, preclinical work, the dog vaccine is behind us. And so you'd start with human development, some preclinical data and clinical trials, as I've described. Optimistically, this could cost about $300 million or so. That's still pretty modest from that in today's world. And so I think the beauty here is this progress from mouse to dogs, a clear model of the disease in humans, natural disease in humans, and a fairly clear path forward with a lot of challenges, but I think all of them are addressable. And I, personally think the time has come to address this. It's a neglected disease in the world's richest country. We have a live attenuated vaccine candidate. It's the most likely way to elicit immune response resembling natural infection. It seems to be safe. It's a relatively low technical risk, which has been mitigated by the dog work. I think the manufacturing control aspects are feasible. We've looked at the market size. We think it's regional, but we think potentially profitable, which should bring in some private money, but it's certainly going to require public funding and incentives. The PRV as one way, it was turned down by the FDA, but one try, but as Winston Churchill said, keep trying. And I think private money will follow once the path is set. So thank you for your attention. That's all I have.